So, you want to return a sample of extraterrestrial life to the Earth. You might be astonished at their complex requirements. Your first thought might be, why not just return it to a glove box in a biohazard level 4 facility? We know how to handle hazardous biological materials, and that's what the experts thought too back in 1978. But actually, they would return it to a quarantine facility in low Earth orbit called Anteus. But a similar idea, just glove boxes. However, soon these ideas began to seem far too simple. First came the discovery of ultra-microbacteria. The ones shown on the left in the slide are taken from narrow liquid veins at depths of over 3 kilometers in a Greenland glacier. They are of special interest for Europa as they can use ammonium chloride as a nitrogen source. Strikingly, they can pass through a 0.2 micron filter, such as is used to purify water. 0.2 microns is also the resolution limit for diffraction-limited optical microscopes. You can only see them clearly with an electron microscope. So that's a bit more of a challenge already. I would suggest bypassing all this with an open-ended approach, starting with a telerobotic facility in orbit and only returning to Earth if you know exactly what's in it and what precautions to take. But first, let's continue and see what the problem is with the sample return to Earth. So next came the announcement of fossil Martian nanobes in a meteorite, far smaller than ultramicrobacteria. This soon became controversial and the debate about whether it is life or non-life continues. Anyway, with that background, they held a 1999 workshop to try to figure out how small early life could be. The main problem with making modern cells small is that they need to use ribosomes for catalyzing cell processes. These are huge molecules, 20 nanometers in diameter upwards. They are made of RNA, shown in blue in this diagram, folded together with proteins. If you had, say, a 50 nanometer cell, you can't fit in many 20 nanometer ribosomes, and to translate DNA to RNA, you also need the bulky DNA polymerase and many other things. So the ultramicrobacteria at 200 nanometers are probably as small as modern Earth life can get, apart from viruses, of course. Viruses can be smaller because they don't need any cell machinery but hijack the biochemistry of the host cell. But what if we have only one information polymer, such as RNA? Or could it be PNA, TNA, etc., some other XNA? That would simplify it hugely, as you can drop all the bulky DNA to RNA translation machinery. After doing that, the RNA still needs to catalyze its own replication and form enzymes for other cell processes. So you still need something to take the place of the ribosomes, and for a while they were stuck, because RNA is not as bendy as a protein. So how could it make these complex shapes? But then they discovered that nature's found a way already, the ribozymes. These aren't folded, they consist of lots of RNA fragments joined together, and not even sequential RNA, but from different places in the strand. And these are as tiny as a few nanometers in scale. So now you can pack a 50 nanometer cell with 1,000 ribozymes. With redundancy, that could be 100 different functions. So potentially we now have a robust, complex cell. We don't know how to do this, of course, but you can also argue that the ribosomes and cell machinery of the modern cell is just far too complex to arise spontaneously in one go from non-living precursor cells. So there must have been less complex early life. So how small can a cell like this get, based on RNA and ribozymes in theory? They estimated the smallest you could still call alive would be maybe 5 genes, and that could be 80 nanometers in diameter including the wall, if spherical, and if elongated, only 10 nanometers in diameter, with an internal diameter just 2 nanometers, just wide enough for the ribozymes. The result, by coincidence or otherwise, is similar in size to the larger Martian nanobes. Then we also need to look at horizontal gene transfer. It's at least possible that Enceladus and Europa ocean life is related to us via meteorite from early Earth, or both seeded by life around another star. Here, researchers have found bags of seawater collected from many coastal environments with whatever microbes happen to be in them. They added a gene transfer agent engineered to give antibiotic resistance, because that's easy to detect. By the next day, 47% were antibiotic resistant. They had no reason to need this capability. It just took it up from the GTAs anyway. Here we are talking about microbes that are far more distantly related from each other than animals are from plants. This mechanism of gene transfer must be very ancient, as it is common to all Earth life. So, if we have a common ancestor with Enceladus or Europa life, there is a decent chance that we could share capabilities with this life via GTAs. The GTAs are tiny, of order tens of nanometers in scale again. They consist of a fragment of a gene and a protein coating. So what is the result of these sample return studies? First, we have the unusual requirement to both contain life and also simultaneously exclude even a single amino acid from getting in. This needs a new type of facility, they said, with positive pressure in both directions, outwards and inwards, and so probably double-walled. It should be designed to contain particles as small as 10 nanometers in diameter, like the early life nanos or the GTAs. If that was not possible, a 50 nanometer particle, they said, must not escape under any circumstances. That's your 1000 ribozyme cell. A decision to increase above 10 nanometers, they said, would need a new review by an independent group of experts. They also said that their results would need to be reviewed in the future based on new research. I'm just sharing thoughts here, but recent developments in synthetic minimal cells and XNA biology might play a significant role in the next sample return study. Then they recommended that staff have to be trained and facility ready to run several years before the launch of the return mission. That's to deal with issues of human errors and the novelty of the facility. This was one of the findings with the Apollo sample returns, that there were many lapses in protocol showing the need for a longer period of training before a sample return. Also, they highlighted the need to respond rapidly to any unintended release. Here is an example design, with everything handled by telerobots, with no humans inside. These facilities might well cost $500 million upwards, and might take around 12 years from initial planning to reach the point where it is ready for sample return launch. You have to complete it before you launch the mission that's going to return the sample. All of this is to return just less than a kilogram of material in the case of the successor to Curiosity 2020. It's got somewhat beyond the early ideas of a simple glove box now. Then Margaret Race of SETI did a study of the legal processes involved, and the timescale is rather similar, 10 years or so or more. Back at the time of Apollo 11, equivalent legislation went through in one day. They published quarantine regulations on the day of the launch. It was supposed to be full public debate, but nobody got to check it or change it. That would be totally unacceptable nowadays. So things have changed a lot since then. Carl Sagan was dismayed that the returning astronauts just got out of the capsule in their decontamination suits into an open dinghy. Remember, lunar dust got everywhere inside and on their clothes. Most likely some dust got into the ocean at this point. You would hardly find a worse place to contaminate with extraterrestrial life than the sea. Why did they do this, one wonders? Well, they had a last-minute problem with the crane that was going to pick the capsule out of the water. Rather than leave astronauts to get seasick while they fixed it, mission planners took them out right away in a dinghy. It's not surprising that they did this, but it shows how humans tend to react when there are tiny probabilities involved, even if it's a tiny probability of a major hazard. More immediate concerns take priority. I think we should treat the Apollo precautions as almost entirely symbolic after that. It showed that we cared, but by the end of the 19th century they already knew the moon didn't have an atmosphere. So it was a precaution against something that most people thought wouldn't happen. And with the science we have now, there's nothing they could have done then anyway, as they didn't even know about many of the potential risks, like the ultramicrobacteria, GTAs, or the possibility of extraterrestrial 50 nanometer nanobes. We were just lucky that the moon had no hazardous life on it. So in short, the quarantine regulations of Apollo had no public review, were largely symbolic, were based on now outdated science, and as Margaret Race also points out, they have been rescinded. We have to make a fresh start. So So that's the first thing that's needed by way of legislation to clarify the quarantine laws, which would include legislation for what you do in case a staff member is contaminated due to mistakes made inside the facility. This could also involve consideration of the legislation on human rights, she points out. Is it legally possible to make an arrangement to keep someone in a facility for such reasons, even if they volunteered to do so? Then she looked at the US domestic laws. The process starts with a formal environment impact statement and public hearings, which take several years. Then after that, the presidential directive NSC 25 requires investigation of any alleged impacts on the environment, and this has to follow after all the other legislation is completed, and takes another several years, ending with presidential approval. Then you have numerous international laws on environment protection and health that didn't exist in the Apollo era, and then quite probably other countries' domestic laws would be relevant too, in case of a scenario that impacts on their country during the sample return. So for instance, if the ESA returned the sample, say to France, then US might well still have to pass all this domestic legislation, since potentially this sample return could impact on them. Then there are other things not covered by the Mars sample return studies. What if the sample return capsule is damaged, as happened to the Genesis probe shown in this slide? This was not a back contamination hazard, since it just sampled the solar wind. But in a sample return from Enceladus or Europa, then we must be sure the container can't be breached under any circumstances. That includes also checks to make sure it wasn't breached by a micrometeorite in transit. Then there are other human factors that are hard to quantify, including overall mission design errors, crash and recovery vehicle, various human errors, and accidental or deliberate release later on. He touched on these issues in his study, but just to say that it was not in their remit to consider them. You might wonder, what about the sample returns we've done already from comets and asteroids? Though it's a minority view, some think that there might be a possibility of viable life in them. This shows the Hayabusa asteroid sample return, which broke up in the atmosphere and crashed, but the sample was from an asteroid not expected to include life or life's precursors. This is the planetary protection assessment for its successor, Hayabusa 2, which is due to visit a large, slightly less than 1 kilometre scale carbonaceous chondrite meteorite in July 2018. This is unprocessed material from the early solar system rich in organics, which may have had liquid water originally, so potentially habitable in the past. As you see, they use decision trees, and as you pass down the tree they conclude that there is some possibility of life from freshly excavated material. But they were able to pass it for planetary protection because we get something like 40 tons of asteroid materials every year on the Earth, like this Perseid meteorite, and some of them will be carbonaceous chondrites. The interior of a meteorite typically is very cold, with the coldness of interstellar space, and stays cold during interplanetary space, and stays cold during its passage through the atmosphere when the crust is melted. And smaller ones land gently with low terminal velocity, like a mouse falling off a skyscraper. So whatever there is in them, whether life or not, we are clearly evolved to be adapted to it. So this passed with no special requirements needed. This is called the natural contamination standard, that if the mission involves something that happens naturally, then we don't need to do anything special to protect Earth. Similarly, when geologists unearth ancient water billions of years old, or drill into an old ice sheet, we can use the same standard that this is something that happens due to natural processes. So it is not of concern for planetary protection unless it is a very rare occurrence. However, when it comes to Europa and Enceladus, deep in the gravity wells of their parent gas giants, there's almost zero chance of a meteorite making it all the way back to Earth at the present time. And in any case, it's extremely unlikely that life adapted to deep subsurface ocean could survive transit on a meteorite from these icy moons to Earth. So there's no natural influx, and we have to take stringent protection measures. Most of the discussions for Mars sample return has been on the effect of microbes on humans. This is Joshua Lederberg's assessment that the microbes would not be adapted to infect us, but on the other hand, our immune system would not be adapted to recognize them. If they didn't produce the carbohydrates and peptides typical of Earth life, then they would essentially be invisible to our bodies, treated as if they were part of our body, like an artificial heart. But of course, it's not just humans that are at risk. If it impacted on other life forms, then that could be an issue too. First, there's the idea that XNA-based life could be better than Earth life. For instance, it could be an all-round better metabolism, or more efficient, so you get a smaller cell able to do the same things. Or it could be that, for instance, it is better at photosynthesis. You might well wonder how life in a dark ocean developed photosynthesis. Well, it turns out one form of sulfur bacteria, not the one shown here, has developed the ability to photosynthesize with infrared light from a hydrothermal vent. We have three main forms of photosynthesis on Earth: the type used by plants with variations, the type shown with the green sulfur bacteria shown here, which doesn't produce oxygen and uses hydrogen sulfide in place of water, and then the method used by the haloarchaea, which turns the Red Sea red, where it converts light directly to electric potential by a similar process to the way we see light, and doesn't produce oxygen or sulfur dioxide or anything like that. So what if life on Enceladus uses another method, call it photosynthesis Mark IV? And what if that is more efficient than any of those that we have on Earth? A photosynthetic life form like that, once it adapted to Earth conditions, could spread exponentially, slowly at first, and then gradually faster and faster as in an exponential, and potentially, in the worst case scenario, outcompete all other forms of photosynthesis on Earth. Then there's the idea of XNA-based life, creating chemicals that confuse our biochemistry. In this case, a small molecule created by green algae happens to closely resemble an amino acid in our bodies and gets misincorporated into proteins and may cause folding disorders like Alzheimer's. This is one of many reasons why you shouldn't eat seafood from an algae bloom. There's no advantage to the algae to give humans Alzheimer's. It's just an accidental side effect. With an alien biochemistry, as well as being inedible, it might easily create various small chemicals that either confuse our biochemistry or get misincorporated by our bodies, or in various ways are poisonous to us. This shows another possibility. This is a small, harmless, short-horned grasshopper. It just hops about doing no one any harm. You can easily imagine if you brought back something as harmless seeming as this, that would seem totally innocuous. But every few years, due to some trick or other, it turns into this. A plague of locusts. So why not bypass all this and return our sample to orbit? You would probably think immediately about returning to the ISS, or a purpose-built human facility. However, I'm going to suggest that just cannot work. First, it's not all about humans. What if our trees or grass or cows or sheep or whatever are impacted? We can't take them all up into space to test them in the facility. Then Carl Sagan, you may not know that as well as a science presenter, he was a biologist and one of the first astrobiologists. He raised the vexing question of the latency period. That's the time it takes before the parasite does anything. For a known hazard like rabies, it's simple. If your stock still doesn't have rabies after 4 months, in the case of the most recent UK regulations, then it probably doesn't have it. In the case of leprosy, this period can be several decades. We saw that Apollo quarantine regulations were not given public review. If they were, biologists might have said that they have to stay in there for decades if you want to be sure, rather than the 3 weeks, the largely symbolic period that was used. And in any case, that's a potential violation of human rights. As Margaret Race touched on in her paper, even if they agreed to it, it's not clear you could legally hold them to that. And anyway, imagine if one of the astronauts became seriously sick. We saw how the mission planners reacted just to the prospect of seasickness. A seriously sick astronaut would have been removed from the facility immediately and taken to a hospital. They'd say, we did our bit, but now they're in danger, so the astronauts have priority. So I don't see that working at all. There's also the idea of returning to a facility with no humans on board and leaving it there for a while before returning to Earth. That's another form of quarantine. But then the life might be dormant in whatever conditions we create for it and wake up later on Earth. It might also start off requiring restricted habitats, for instance, strongly alkaline soda-lake conditions and evolved the ability to live more widely after arrival on Earth, decades later. So in summary, human quarantine is just symbolic, unless you know what it is you're quarantining against and why. Quarantine tests involving other life forms from Earth tell us little. There's no substitute for knowing what it is before you let it anywhere near to either humans or Earth itself. So that leads to my suggestion. First, study in orbit with telerobotics. So far, we have never studied anything in space in this way. This is partly due to the time delay and mainly to do with the bandwidth. The bandwidth for our robotic explorers is so small that typically they send commands for the entire day and rely on the information returned to make decisions for the next day. It could be as far away as Pluto or the Kuiper Belt, and it would probably take a comparable amount of time to do the exploration and studies. With a better connection than that, the technology is very advanced now. This shows the Da Vinci system of telerobotic surgery. The surgeon looks into the machine and manipulates instruments with his hands while the patient lies over there to his left. This permits more precise surgery with delicate tools and is used a fair bit nowadays. He could as easily operate from his office or somewhere else entirely, except that for safety reasons he is required to be in the same room. And actually, we have had tests of telerobotic surgery done from a distance. A patient in France was operated on in this way by a surgeon in the States. He had a team of doctors on hand in France, ready to step in if necessary, but they were not needed and the operation was a success. It's not widely known, but the Japanese are planning to build a large radio telescope in orbit assembled by telerobotic methods and have already done tests in orbit that show it can be done successfully. So, on to my idea. Our aim is to get our capsule to above GEO orbit as the safest place to do the telerobotic studies. The idea is to use a similar approach to the NASA asteroid return mission plans. Return the sample to the Earth L2 first, where the Earth and Sun's gravity pulls are in a balance, shown here as EL2. Once we've got that far, there's an almost zero delta V transfer to the lunar L1, and we are well into cislunar space. When coming in from Jupiter, it's not so easy to get captured by the Earth-Moon system, however. First, we must always bias our trajectory away from Earth, so that the failure can never lead to impact. This rules out close flybys of Earth using atmospheric drag or the Orbit effect, the effect which the Indian Moon mission used in the other direction to get to Mars with minimum of fuel. And you may not be able to do it all via gravity assist either. Perhaps 2 to 3 km a second of extra delta V is needed to get the capsule into Earth orbit, similar to Rosetta's escape velocity when it first left the vicinity of the Earth. Experts would need to sort out the techy details here about how the return vehicle could achieve this, so this is just touching on something that would have to be sorted out. Once you get to the lunar L1, it's easy to get into a distant retrograde orbit around the Moon. This is actually an elliptical orbit around the Earth, in the same direction as the Moon, so it's prograde around the Earth. But when the satellite is closer to Earth, it goes around faster than the Moon, and when further away, it goes around the Earth more slowly, and the combined effect is that when seen from the Moon, it seems to be orbiting in a retrograde direction. It can orbit either inside or outside of the lunar L1 and L2 positions, and one example is shown here. The idea is that your sample return mission can get this far without any assistance from Earth. Then it would be in a reasonably stable orbit, at least for 100 years. So there's no hurry for the next step. But you would then send an unmanned spacecraft from Earth to capture the capsule. This is actually the way that some sample return proposals work, with the spacecraft returning to Earth. But instead, you return the sample to a high orbit. This would be the start of your telerobotic facility. Then, the natural place to take it to is above GEO, say 5,000 kilometers above, to be well away from geostationary satellites and their graveyard orbit. That's over 1 km per second delta V needed to hit the Moon, and it's also over 1 km per second delta V to get to a geostationary transfer orbit to hit the Earth. So it is now as safe as it can possibly be in cislunar space. This is not going to happen until at least a decade or two from now, so we probably have the capability to send 100 tons or more in one go to above GEO by then. So it's easy to send large machines up there to study it. And we're doing this, of course, for benefit of all humanity, as required in the Outer Space Treaty. So individual countries, such as say India or Japan, could mount their own expeditions to our new facility as part of a joint international effort. The only requirement is that all materials are transferred only one way to the facility unless thoroughly sterilised. You could return some small samples to Earth right away if they are sterilised 100% first for the few experiments that can't be done in orbit, such as to put them into the paths of particle accelerators. Then it might be that you soon find that there's nothing there except amino acids and maybe autopoietic cells, but no life, and everything is so feeble it would be eaten easily by Earth life. If totally sure, as a consensus amongst all astrobiologists and after public debate, you could return it to Earth. In this case, there's no need for a special facility or all those new laws, because you know there's no life in it, or no hazardous life. On the other hand, if you find an exotic XNA-based life, say with a more efficient metabolism than Earth life, everyone would agree that we have to take extreme care. And in that case, you might not return it for a considerable period of time, except possibly sterilised samples. And if you did, everyone would understand the need for an elaborate facility to contain it, and would take great care, and you wouldn't have concerns about breaches of protocol due to natural human inattention when you think there's only a minute chance that there's anything to protect against. So this is an open-ended approach, where at each stage you know the implications of what you're doing, and don't have to rely on making probability assessments that really exobiologists haven't got anything like sufficient data to provide. And you don't have to build a facility on Earth that has to contain all conceivable possibilities for alien microbes. You just have to contain the ones that you already know about and have studied carefully. So you, at each stage, you could, when you first return it to above GEO, geostationary orbit, then at that stage, if you inspect it and you find there's no life there already, you just sterilise it and return it to Earth. And then, if not, you start inspecting it and looking at it carefully. You may send up more machines from the Earth and other modules and things if necessary, and then eventually when you do bring it back to Earth, you only build as much of a facility as you need for whatever it is that you're bringing back. And so at each stage you only do as much as is needed at that stage, and with any luck you can go all the way through without having to introduce any new laws as well. So that's my suggestion.